I wish I could get just like a super... I'm not gonna be able to find like an explanation of this without it being like a crazy conspiracy thing.
So,
like, I don't know if this is somebody argue. Like, I just want like an explanation of, did we look at this and were they... Fuck it.
All right, we've hit 1203. And
That guy is correct, though. Look up genetic drift and genetic shift. That has nothing to do with what we're talking about. RNA is much less stable than DNA. I don't think that has much to do with what we're talking about. And as a result, can have lots of random cuts and insertions. But we're not talking about a random cut or insertion, right?
And we're starting to level
Although,
up.
like, technically all mutations are random, but... Okay,
So
I don't
I
care.
guess we'll
All
get
right.
going.
All right, this is going to be boring. Get ready.
Hello, everybody, and welcome to this week's SP Grid webinar series. Today we have two presenters who are going to talk about some viral-related stuff, which is always interesting, and particularly interesting with the current events over the past few months. If you have questions, you can send them to me or one of the other hosts in chat, and we'll moderate those at the end. And so for today, as I said, we have two speakers, both from the Ashari Lab at Duke University School of Medicine. So thank you for joining us. Sophie Goebel and Kartik Mani, and we will start off with Sophie. So with that, feel free to take it away.
great. Let me share my screen. Okay, so you can see my screen.
Looks
Oh
good.
my God, there it
Okay,
is.
great. So today I'll be talking about the work I've been doing since the start of the pandemic. Actually, I joined Pimbara's lab right at the start of the pandemic. So we are studying how the SARS-CoV-2 spike D614G mutation, how a single amino acid change makes a big difference in the structure and particularly susceptibility of the spike protein. So I'm happy to say that this work that I'll be talking about today has just been submitted to bioRxiv and is available since last night. So
Our preprint server. Oh boy.
I'm inviting you to actually go see all the whole paper. So first, well, I don't think I need really a big introduction for the COVID-19 pandemic. We all know how bad it's been with more than 1 million deaths worldwide, 37 million cases. The United States have been pretty badly affected, but it's all over the world. So that prompted us to really study the proteins and understand how to respond to this global crisis. So beta coronavirus are divided in different clades. There's different beta coronavirus that have been known to infect humans. Actually, there's 7 of them. Of the 7 viruses, there's only 3 that have been responsible for the bad epidemics. So the SARS virus, the MERS virus, and the SARS-CoV-2 that we currently are fighting against. And so as you can see, they're divided in different clades. So the alpha, beta, and gamma coronavirus, and the SARS, MERS, and SARS-CoV-2 are all beta coronavirus. So why are they called beta coronavirus is due to the fact on their surface, there's the corona, which is the Latin word for crown, which is made by the spike protein that you can see here, which is an homoterameric glycoprotein at the surface of the virus. And that protein binds to the angiotensin converting enzyme 2 receptors, or ACE2, using the receptor binding domain at the top of the spike. So today I'll drive you through this image, which is, I think, the main figure of the paper. There's 3 different sections of the talk. So the first one, I'll discuss the K986 and V987 proline mutation, which are stabilizing mutations that were introduced in the spike ectodomain construct. I'll then go through the D614G mutation that has been attracting a lot of attention due to the fact that this mutation became highly predominant really fast through the pandemic. And finally, I'll discuss also the Furin cleavage site, which is important for the activation of the spike for the infection to the human. So as I said, the first part is going to be on the construct, the ectodomain soluble construct that is currently used, which is called the SGSAS-PP. It's called like this because the Furin site is mutated to, which is RRAR in the native protein, is mutated to SGSAS to actually make the construct not cleavable while expressing it in cells. And there's also 2 stabilizing mutations, which are K986 and Val987, change for prolines. This is introduced based on observation of the MERS and SARS spike protein.
When it talks here about this evolution, what is it evolving from? Like, was this all observed in the same like SARS-CoV-2 thing, or are they guessing like what it looked like? Like, so when they before this amino acid mutation, was it still SARS-CoV-2, or?
That without those mutations, we're showing a lot of the fusion conformation. So there was a lot of heterogeneity in the prep of the spike protein. Sorry. And by introducing those 2 prolines, it was shown that the heterogeneity was reduced and the construct were really the fusion conformation. So the SARS-CoV-2, those two proteins were introduced in the SARS-CoV-2 construct based on the MERS and SARS. So we first asked, do we really need those two stabilizing mutations, or we could remove them and the SARS-CoV-2 protein would remain still in its pre-fusion conformation? So throughout the talk, I'll be using three main biophysical methods to measure how the protein is behaving. So I'll use the size exclusion chromatography, which is a way of separating protein based on their mass without denaturating the protein. I'll also be talking of SDS-based gel denaturating type of gel.
Is it really denaturating, not denaturing?
Okay. So basically separating the trimers, the monomers of the trimer, and you can see again it separates the protein based on their mass. And finally, I'll be talking also of differential scanning fluorimetry, or DSF, which is a way of measuring the thermal stability of the protein by increasing the temperature.
It's the difference between ancestral SARS and SARS-CoV-2. But this mutant was made in modern SARS-CoV-2 to explain the evolution. Okay.
The protein denaturate and expose the tryptophan.
When is it considered like the next, like SARS-CoV-3 or SARS-CoV-4? What has to change for it to be updated on the version number?
And aromatic residues, and we're measuring the intrinsic fluorescence of the protein throughout the thermal denaturation of it. So comparing the SGSAS PP versus the SGSAS protein, we can see that they're really similar proteins. They elute on similarly on the size exclusion chromatography. They have a similar pattern of bands on the SDS-based gel, and their thermal denaturation profiles is also really similar. So knowing that the MERS and SARS were showing a lot of differences in their NSEM showing fusion conformation, we then did some negative spin electron microscopy. Here I'm showing the raw micrograph. We did 2D classification, and as you can see here, we're mostly seeing a nice form spike, nice prefusion conformation of this spike, suggesting that the prolines are not necessarily needed in that case for maintaining the SARS-CoV-2 tumeric conformation. So we then ask, is the antigenicity affected? So I'll be showing two different types of assays where we add either the spike bound to the surface or the spike flowing over an antibody that is bound to the surface, just to make sure that there's no issues due to the system. So that's basically a way of controlling here. And what we've seen is that both the GSAS and the SGSAS PP construct really behave similarly in terms of binding to different proteins. So we have ACE2 and CR3022, which requires an RBD up conformation. We have a glycan antibody, which is called 2G12, which binds to an S2 site in the site of the S2 domain in the spike protein. And we have also two convalescent patient antibodies, AB511 and 712, which are binding respectively to the S2 domain of the protein or the S2 site. So the binding site of ACE2 at the top of the spike. So knowing that both proteins were behaving similarly, we ask things like 11 protein,
what is the glycan thing? Hold on.
Which requires an RBD up conformation. We have a glycan antibody, which is called 2G12, which binds to an S2 site.
Okay, glycan is a type of antibody? Okay.
In the site of the S2 domain and the spike protein. And we have also two convalescent patient antibodies, AB511 and 712, which are binding respectively to the S2 domain of the protein or the S2 site. So the binding site of ACE2 at the top of the spike. So knowing that both proteins are behaving similarly, we ask, is it possible, can we see the structural details? Can we see how the protein is different or exactly similar basically to the SGSAS PP? How does the 2PP affect the structure of the protein? So we obtained the Cryo-EM structures. Jesus. So you can see here on the side the different N-terminal domains, so the NTDs, there's 3 of them, and on the top the 3 RBD domains, which makes the interaction with the ACE2 receptor. Here is the top view of the spike. And here you can see the RBD going up in the 1-up conformation. So in that data set, we were able to identify both 1-up and 3-down conformation. What we found is that the 3-down conformation, the particles were more predominant compared to the 1-up conformation. And overall, both structures are showing really high, really similar aromacities compared to the structures that were already available in the PDB. So with those structures, we went in and tried to see how this lysine valine mutation to 2 prolines made a difference or not. And what we've seen is that if you mutate lysine 986 to proline, you might in fact affect a potential solid bridge between residue D and impact it with residue D427 because the proline doesn't allow for the solid bridge. So overall, the structures are really similar, but there might be a little bit more interactions in the non-PP version that are possible that we're not seeing in the PP version.
Of course.
So next, as I said earlier, the D614G mutation has been really quickly throughout the pandemic overwhelming all the sequencing data that was coming from patients. So as you can see, March 1st, we had mostly the D version, which is in yellow. And mid-March, the blue version, which is the G614, already became more predominant. And by the end of April, it was the main strain isolated. This was identified by Betty Korber and David Montefiore. And so we asked, what does this mutation change in terms of the structure of the protein? So as I said, the D614G mutation is actually making potential hydrogen bonds with interprotomer hydrogen bonds, implicating threonine 859 of the next protomer. And it was also hypothesized to impact glycosylation at a nearby site in 616. So we first produced the protein in the context without the two proteins mutation. So throughout this talk, I'll be using now the SGSAS context.
Okay, this is not going to help us. Furin cleavage sites naturally occur in coronaviruses. Abstract. I need like something between like a bachelor's and between like a fucking professional research scientist. Okay, the coronavirus disease 2019, COVID-19 pandemic caused by coronavirus SARS-CoV-2 is still rapidly spreading. Scientists all around the world are studying the infection process of the virus and looking for therapeutic solutions to prevent and cure the disease. Spike protein is one of the most important targets in COVID-19 research, not only in mechanism study, but also in vaccine development and therapeutic antibody design. The spike protein forms a homotrimer, got 'em, and protrudes outside the membrane of the virion. It binds to angiotensin-converting enzyme 2 on human cell surfaces, undergoes conformational changes, and mediates the fusion of the virion to human cells. Figure 1, structure of SARS-CoV-2 spike protein. A, spike homotrimer and open conformation. B, cleavage sites on spike protein marked by arrows. Red cartoon, S1 is light and dark blue cartoon. S2 equals dark blue cartoon. Okay. For interpretation of the references to color in this figure legend, the reader is referred to the web version of the article. Okay. The spike protein consists of two subunits, the N-terminal S1 subunit containing the receptor binding domain that interacts with ACE2 and induces conformational change. What does that mean? Conformational change. Does it cause it to morph once it gets inside the thing? Is that what that means? A conformational change is a change in the three-dimensional shape or structure of a molecule, most commonly a protein or nucleic acid, due to environmental factors or binding interactions. Okay. Okay, so S1 is responsible for binding to ACE2 and then having the protein refold itself afterwards. And then while the C terminal S2 subunit containing the fusion peptide that is responsible for fusing the membranes of virion in human cells to function properly, the spike protein needs to be cleaved by host proteases to separate the two subunits. Study of SARS-CoV in 2002-2003 and belonging to the same species as SARS-CoV-2 showed that cleavage at S1 and S2 enhances fusogenicity of spike protein. How genetically fusible it is, or what? Facilitating fusion, especially relating to cells. The incorporation of certain constituent lipids into a liposome. Okay, how easily it's fused into a cell, I guess. Enhances fusogenicity of spike protein. Another cleavage site producing S2 fusion peptide in the C-terminal region of S2 was also identified. The S2 site is not far from the S1 S2 site. And cleavage either of them or both of them can yield, or if you cleave either or both of them, can yield the separation of two subunits of spike. For coronaviruses, multiple proteases were reported responsible for the cleavage, including TMPRSS2 and cathepsin, CTSL, and trypsin. Okay. And these are just enzymes that float around in cells, basically. Right? May augment virus entry into airway epithelium, increasing virus propagation by cleaving the HA protein of the influenza cell. Okay. The spike protein of MERS-CoV was discovered to be more effectively cleaved by another protease, Furin. In MERS-CoV, both S1, S2, and S2 sites have sequence RXXR, the Furin recognition motif. In the human body, Furin is ubiquitously expressed. When it says ubiquitously, that means all cells have this enzyme, or this protease, I should say. This may explain the highly lethal nature and high rate of multiple system failure caused by MERS. Surprisingly, SARS-CoV-2 has the Furin recognition motif at S1, S2, causing, by a 12 nucleotide insertion, not presented even of its closest relatives. This stimulates a conspiracy that this Furin site can only be manual work, thus SARS-CoV-2 must be created in laboratory. Here we analyze the sequences of coronaviruses and found Furin sites occurred independently from multiple times during evolution. This exhibits natural occurrence of Furin cleavage sites in SARS-CoV-2 spike protein is highly possible. Thus, the insertion of Furin cleavage site into SARS-CoV-2 spike protein is not necessarily a result of manual work. Wait, hold on. More effectively. So what is the part of the S1 or S2 subunit? What is the part of the spike protein that makes it so much more likely to infect epithelial cells? That's tissue of the... I horribly spelled it incorrectly. No. Relating to or denoting the thin tissues forming the outer layer of a body's surface and lining the alimentary canal and other hollow structures that line the internal and external surfaces of the body. Are these more likely to be like in the lungs? It's like a lining of an origin. It's the S1, S2 boundary between the two matrix, which is the Furin cleavage site. Okay, wait, I don't understand what this means then. Wait. Hold on. In the human body, Furin is ubiquitously expressed. This may explain the highly lethal nature and high rate of multiple system failure caused by MERS. But it sounds like this Furin protease is in all of your cells, right? Pretty much yes. Okay. So then it sounds like one of the convenient things for SARS-CoV-2. So in MERS-CoV, there is a Furin cleavage site at both S1, S2, and the S2 sites, meaning that every single cell in your body, once it admits the MERS-CoV virus, it cleaves here, and then it cleaves here, so that every cell in your body can become infected. But for SARS-CoV-2, there was only one Furin cleavage site at the S1, S2 boundary, and then something else cleaves it at the S2 site? Wait, Furin is in all cells, but ACE is not. fuck it. I'll just keep reading it. Save this helps. Fuck. We need like a whole like sub course on this bullshit. We map the Furin recognition motif RXXR at both S1, S2, and S2 primes at positions in phylogenetic trees of spike protein sequences in the Saber virus plus Hybe-covirus plus, or Saber Sarb-covirus. I don't know. Furin cleavage sites at either position occur only in limited ranges. Strains of SARS-CoV-2, we also added sequences from the GISAID database, have Furin cleavage sites at spike S1, S2. Moreover, SARS-CoV-2 is the only virus in subgenus Sarb-covirus having this feature, while even its closest relatives, bat coronavirus and pangolin coronavirus, do not have a Furin site. However, in Hybe-covirus, the sister clade of Sarb-covirus, a Hippocyterus has a Furin site at S1, S2. Interestingly, the other member in Hybe-covirus lacks such sites. SARS-CoV-2 strains are not sister groups. Okay. The S1 domain is what helps the spike protein recognize and bind ACE2. S2 is the thing that drives fusion viral entry. The Furin site allows the spike to become active. I understand that. I'm just trying to figure out what was the part that makes this target... what's the thing that makes this target like the lungs? Is it just the ACE2 part? What did MERS bind to? Oh, maybe that's it. Are your ACE2 receptors, are those normally only in your anti-angio converting enzyme 2? ACE2 is known to be expressed in various human organs, and its organ and cell-specific expression suggests a major role in the regulation of cardiovascular and renal function as well as fertility. Ooh, yeah. Functional receptor for the spike glycoprotein in human... okay. All right, so the ACE2 part... gotcha. Okay. Sorry, Jesus. Okay, that helps a little. Okay. It's the ACE2 part that that is unique to SARS-CoV-2, or maybe not unique to it, but I don't think the MERS... Well, can we just look this up? MERS virus Wikipedia. How do you get into the cell? Virology, transmission, single-stranded RNA belonging to the genus beta. Oh, that didn't help. What is... Fuck, shouldn't it be here? Do I need this? MERS-related coronavirus. Terms of phenomenon indicating the growth or turning movement of an organism. Is it in here? Am I retarded? How does it get into the cell? Oh, I am retarded. The infecting virus is an enveloped positive sense single-strand RNA virus which enters its host cell by binding to the DPP4 receptor. Okay. So a different receptor, CD26, is a protein that in humans is encoded by the DPP4 gene, which is what this is found more commonly everywhere in the body. Is an enzyme expressed on the surface of most cells, and is associated with immune regulation, signal transduction, and apoptosis. Okay, so the reason why MERS was all over the body more was because this gene, the DPP4 gene, is in basically all the cells, or is activated in all the cells, and in a coded form basically all the cells. So MERS will infect every single cell in the body For SARS-CoV-2, its gate, and that's because it uses this to get into the cell. For SARS-CoV-2, it uses the ACE2, that enzyme, to get into the cell. Both of them require or use furin to cleave their entry points. I don't know if it's called a spike protein on both. So the furin protease is found throughout your entire body, but the ACE2 gateway is more likely to be found on epithelial cells or cells that are lining shit, so like in your lungs or whatever. Okay. Okay. All right. Okay, that makes more sense, I think. Foreign cleavage is either position. Okay. Do we care about anything else in here? I don't know if we do. Holy shit. Coronavirus with spike containing furin cleavage site may have advantage in spreading. Deletions of furin cleavage sites at SARS-CoV-2 attenuates replication on respiratory cells and pathogenesis in hamsters. Damn. Furin inhibitors suppress viral production and cytopathic effects in kidney cells. Natural polymorphisms, losing furin recognition, motif in SARS-CoV-2 spikes, S1, S2 observed, but very rare. Variations in this region are more common in viruses cultured in vitro than viruses isolated from clinical samples, suggesting this cleavage site is under selection pressure in human body. Our analysis exhibits furin cleavage sites at spikes S1, S2 has occurred independently for several times in coronavirus. Consequently, natural occurring at the site in SARS-CoV-2 is highly possible. This is further supported by other observed. Okay, all right, don't care. Okay, Jesus Christ. Okay, I think we're good enough. I think. All right.
To remote bat caves and immunize bats against this very same viruses. So maybe they should have sprayed them with ivermectin. I'm sure Brett Weinstein would support that idea. And my slides are getting a little out of alignment because of this app that doesn't allow me to share the screen. Anyways, yeah, okay, well, basically, the first phase of the DEFUSE proposal was first identifying these dangerous viruses and that pose a threat to humans before actually diffusing them. And they were focusing, this particular proposal was focusing just on SARS-like viruses, the same family that SARS-1 and now SARS-2 comes from. And it's interesting that like either the DEFUSE authors had this incredible foresight about a lurking SARS-2 progenitor that was only missing a furin cleavage site that was waiting to spark a global pandemic, or the other alternative being that DEFUSE was actually a self-fulfilling prophecy. So, I mean, and this is why DEFUSE is so relevant to our discussion, because it outlined not just sampling many novel SARS-like viruses, but also genetically modifying them to model if they could evolve to become more dangerous to humans. And so in particular, they talked about, first of all, looking for furin cleavage sites in these viruses, and then if they didn't find them, basically creating novel furin cleavage sites in SARS-like viruses. And so in the next few slides, just wanted to highlight some key points of that proposal that I thought were particularly relevant. And so, for example, here, just wanted to highlight that they were talking about sequencing full-length genomes of novel viruses, and then also synthesizing them into live viruses, essentially creating novel backbones and turning them into live viruses. This is what they mean by recovery recombinant viruses. And I mean, they had plenty of viruses to study. DEFUSE itself mentioned that they already had 180 sequenced but unpublished SARS-like viruses, and then they talked about including into the analysis of DEFUSE 10,000 samples from 6 Southeast Asian countries, and they planned to do a lot of live virus work at the WIV. And also I wanted to point out that for the screening of the viruses that pose the threat to humans, they plan to look not just at genomic data, but also at computer binding modeling, and also looking at 3D protein structure of the spike trimer. And so some critics of the lab leak hypothesis say that SARS-2 would have been uninteresting because it's only 80% close to SARS-1. But of course, in the diffuse proposal, SHCO14 is like 88% close to the spike. But like besides all that percentages, if you actually do protein modeling, you'll see that the SARS-1 and SARS-2 spikes are actually very close in the protein. And so sometimes you can have like different amino acids in the protein, so like they wouldn't be identical on the protein level, but functionally you can have similar amino acids, and functionally the protein can actually be quite close. And of course we know that SARS-2 is very good at binding the human ACE2 receptor, and even better than SARS-1. So if they did some kind of modeling on the spike, I think that would have been apparent just how potentially dangerous SARS-2 is, or the spike in SARS-2 kind of viruses to humans. Okay, just also I want to point out that while they were...
This gets like very into the weeds, but like I don't know what the answer is or not, but like the issue with that statement is, so he's saying, and I don't know who to rule as being more credible here. So he's saying that there was a change in the receptor binding domain that made it more likely to bind to the ACE2 receptor, but then he kind of offhandedly says if they would have just modeled it, then they could have discovered that. But that original paper says that models don't predict that. I don't know who's right there, though. I literally have no fucking idea. While the analyses above suggest that SARS-CoV-2 may bind human ACE2 with high affinity, computational analyses predict that the interaction is not ideal and that the RBD sequence is different from those shown in SARS-CoV to be optimal for receptor binding. So like in this paper, they claim that wouldn't be. Now, this was an early paper, so maybe the understanding has changed since then, although I don't know how you would change the understanding afterwards and claim that people knew it beforehand. I have no idea how you would evaluate who's right or wrong there. I don't have the background to say.
Okay, just also I want to point out that while they were planning to use chimeric viruses for their initial analysis, they also plan to then validate any interesting results from that analysis with full-length viable viruses, like essentially creating novel backbones, as I already pointed out in the previous slide. And as I said, the live virus work was planned to be done in Wuhan, in the Wuhan Institute of Virology.
I also don't know, well, this is an answer that we could research. Does this mean when you say that you're recovering recombinant viruses using established techniques in genome length RNA and electroporation to recover recombinant viruses, does that mean you're creating novel backbones? Or does that just mean you're trying to track the evolution just in one particular part that you're trying to change? Does recovering recombinant viruses create novel backbones for you to experiment with? I don't know the answer to that. I'm not sure. But I don't know, maybe we'll get into that.
Cleavage site. None of the other 1,500 SARS-like viruses have a furin cleavage site, and so in light of like the intentions outlined in the DEFUSE proposal, it's very, very suspicious. It would grow in the human airway epithelial cell culture. We did so. Also I want to point out that while they were planning to use chimeric viruses for their initial analysis, they also plan to then validate any interesting results from that analysis with full-length viable viruses, like essentially creating novel backbones, as I already pointed out in the previous slide. And as I said, the live virus work was planned to be done in Wuhan, in the Wuhan Institute of Virology.
Recombinant means multiple. What is, I don't know what this means. Recombinant virus? I don't know how. I don't even know if this is a thing. Jesus Christ.
In the pursuit of a cure for AIDS, scientists have devoted great efforts into studying the HIV lentivirus. Although a cure for AIDS remains to be seen, research efforts into the HIV lentivirus have led to major developments of powerful HIV-based tools for transgene delivery. Today, lentiviral vectors are major tools for basic and translational research, enabling scientists to safely overexpress or silence genes, immunize against a range of diseases, generate transgenic organisms, and much more. The lentivirus is part of the retroviridity family of viruses, with HIV-1 being the most widely studied. Members of this family contain a positive single-stranded RNA that is reverse transcribed into DNA and integrated into the host cell's genome. The HIV lentiviral RNA genome contains three major genes, GAG, POL, and N. The GAG gene encodes protective core and matrix proteins for viral assembly and infection. The Pol gene encodes for reverse transcriptase, RNase H, and integrase, enzymes crucial for reverse transcription and genomic integration. The EMF gene encodes for surface glycoproteins that determine tropism and enable entry into the cell. The lentivirus genome also contains two important regulatory genes, TAT and REV, that activate viral transcription, as well as four accessory genes, VIF, VPR, VPU, and NEF, that are less essential for virus replication in host cells.
Cool. I think it's going to tell us.
Lentiviruses have lots of potential as tools for gene delivery and gene therapy. This is because their stable integration into the host cell genome ensures long-term expression for stable cell line generation. They can infect both dividing and non-dividing cells, such as hepatocytes and neurons, cells that are notoriously difficult to infect. They have broad tropism, making them effective vehicles for delivering CRISPR sgRNA constructs to edit the genes within any tissue type. They don't generate immunogenic proteins, and they can deliver transgene fragments as large as 9 kb. Immunogenic proteins, and they can deliver transgene fragments as large as 9 kb.
Does that mean kilobytes, or is that something different? Is that just measuring how many like amino acids or nucleotides or whatever that you could insert into a kilobase? Base, okay.
Because of all these advantages, great efforts have been put into developing recombinant lentiviruses for clinical and research use due to their robust infection ability. However, precautions are needed to ensure replication-competent lentiviruses are not accidentally generated via recombinations between the delivered as well as endogenous viral elements in producer cells. In an effort to reduce biosafety risk, 3 generations of recombinant lentiviral packaging systems have been developed, with the first generation system no longer in use due to biosafety risks. The second generation system splits the essential components of the lentiviral system across 3 plasmids that are delivered separately for safety. The transfer plasmid encodes for the transgene. It also contains cis-acting elements such as a 5' and 3' LTRs essential for promoting RNA polymerase II to begin transcription of viral mRNA, and the size sequence which signals genome packaging. The packaging plasmid is provided in trans and encodes only essential transacting genes, GAG, POL, REV, and TAD, that are required for entry and integration of the viral genome. The envelope plasmid contains genes encoding for envelope proteins. For safety reasons, all other genes involved in viral propagation, for example, VPR, VIF, VPU, and NEF, are excluded. To almost completely eliminate dangerous lentiviral recombination events, an even safer third-generation system was created.
Almost completely, almost.
In this system, the viral genome is split into 4 plasmids. A plasmid containing only the packaging genes, GAG and POL, a plasmid with only the regulatory gene, REV, a plasmid carrying only the envelope gene, ENV, and a transgene plasmid. In addition, the LTRs flanking the transgene are further modified as they contain enhancer and promoter regions that can activate adjacent cellular proto-oncogenes. By removing this enhancer and promoter region, U3, from the 3' LTR, a replication-incompetent, self-inactivating system is created as this deletion is transferred to the 5' LTR after reverse transcription and integration. The U3 of the 5' LTR is also replaced with a CMV promoter to eliminate the need for the transcriptional transactivator, TAT. Although the third generation is significantly safer, its viral yield is typically lower than that of the second generation system. At ABM, we offer both second and third generation lentiviral packaging systems to suit any application. Finally, as tropism is determined by the glycoproteins on the viral surface, a lentivirus's tropism can be further broadened by replacing the HIV-1 EMF glycoprotein with the envelope glycoprotein G from the vesicular stomatitis virus, VSVG. VSVG is incorporated in all ABM lentivirus packaging systems to widen the range of cell types the virus can bind to. Recombinant lentiviral systems show great promise as potent tools for gene therapy to treat HIV infection, cancer, Parkinson's disease, and many more. A breakthrough is surely not far on the horizon. In the meantime, recombinant lentiviral systems have secured their place as an invaluable tool for advancing in vitro and in vivo research across a variety of areas in biology and the life sciences. From siRNA lentivectors to CRISPR-Cas9 lentivectors to chemate-inducible lentivectors, our lentiviral vectors and ready-to-use lentiviruses can be used to manipulate gene expression of any human, mouse, or rat gene. Simply select your gene of interest.
Okay, well, this doesn't really help us at all, but that is really fucking cool. Okay, somebody give me the cheat. What the fuck does recombinant mean? Recombinant virus is basically a virus where you give it an alternative spike so it can act like your virus of interest but not be the actual virus. This means you can study how a SARS-CoV-2 spike works. Oh, recombinant just means it's... Okay, never mind. That makes sense. Okay. So this is a recombinant thing or these packages are recombinant because it's sucking out different parts of the HIV virus, and then it's inserting whatever genetic material it wants you to. So it's a recombinant thing, right? Okay. Why, if you have, wait, I don't understand how wait, what am I thinking of? Does this truly mean that like HIV would never be cured? Ever? That seems fucked. Because it's literally part of your DNA and it infects every fucking cell in your body? Would it ever be possible to cure something like that? Or I guess if you infect something, I don't even know. I have no idea. Well, that sucks. What? How many? Oh no, never mind. There are other viruses that can encode themselves or become integrated into your DNA. But my guess is that most of the viruses that do this will cause the cells to eventually express some sort of protein that your immune system will detect and kill, right? It will generate an immunogenic protein for the vast majority of viruses, I guess, right? But I guess HIV doesn't. Yes, but it's missing the replication machinery, so recombinant viruses only undergo one round of infection. Wait, but but if it infects the DNA of a dividing cell, oh, but it would just be the cell dividing. That wouldn't necessarily mean that it spreads to the rest of your cells, right? Because it's not infecting all of your cells. It's just that cell would divide it or whatever. But okay, interesting. Okay. Jesus, fuck me. All right, back here.
And I think this is probably the most important page in all of DEFUSE, as it talks about the very same sort of like gain of function genetic engineering that they plan to do that I think could lead to SARS-2. So here, like they say, we'll synthesize quasi-species with novel combinations of mutations to determine the effects on specific genetic traits on spillover potential of future and unknown recombinants. And the most striking part here is where they propose engineering novel furin cleavage sites and then testing how these modified viruses would grow in the human airway epithelial cell cultures. And also then seeing how pathogenic they could be to humanized mice, basically mice tailored to have human ACE2 receptors in their lungs. And you know, don't want to like belabor the point, but SARS-2 is unique in its family of like 1,500 known SARS-like viruses, unique in having the furin cleavage site. None of the other 1,500 SARS-like viruses have a furin cleavage site. And so in light of like the intentions...
Was that true? There were other coronaviruses with FCSs, but I don't know about other... I don't know about other SARS-like viruses. I also don't know the validity of this paper. This was published in Stem Cell Research. Is that the name of the journal? MERS has the, yeah, but MERS is not SARS, right? I don't know if MERS is the same, or it's a coronavirus. I guess MERS and SARS aren't really, they're both coronaviruses, right? So it doesn't, there's not, the distinction there is only the disease that it causes, right?
100 SARS-like viruses have a furin cleavage site, and so in light of like the intentions outlined in the DEFUSE proposal, it's very, very suspicious. Oh, and sometimes zoonosis proponents dismiss the DEFUSE suggestion of engineering novel furin cleavage site because they say that was only meant to be done in chimeric viruses. I'm not sure why, but in any case, DEFUSE in a couple of places, like this is the second time I'm having basically pointing this out, that they plan to validate the results from chimeric viruses with in full-length genomes. So basically, they were anyways planning to create novel backbones. They were saying like we will recover 3 to 5 full-length novel viruses per year, but also if chimeric viruses showed some interesting results, then would that lead to them validating those results in the full-length viruses from where the spikes for those chimeras came from. And they would then look for viral growth in primary human airway epithelial cultures and in vivo, and basically in these cell lines, the furin cleavage site, if they inserted it, it would not mutate away. Because in some cell types, like Vero cells, for example, the furin cleavage sites tends to mutate away because Vero cells either don't express furin, they have very low furin expression, so there's like selective pressure against the furin cleavage site. But in human airway epithelial cells, the furin cleavage site would actually remain. The furin cleavage, like selective pressure against the Vero cells, for example, the furin cleavage site mutate away. Because in some cell types, like Vero cells, for example, the furin cleavage sites tends to mutate away because Vero cells either don't express furin, they have
I don't know what a Vero means. I know in vitro and in vivo. I don't know.
Have very low furin expression, so there's like selective pressure against the furin cleavage site. But in human airway epithelial cells, the furin cleavage site would actually remain. And so basically, when someone sees the DEFUSE proposal, I think it becomes very suspicious that the unique features of SARS-2 align so well with what the DEFUSE proposal was talking about. And I mean, this table just highlights some of these key features, like the ACE2 receptor binding. Of course, they were talking about this being the primary screening factor for picking which viruses to work on. And SARS-2 is obviously very good at human ACE2 receptor binding, maybe even like human receptors is the top.
Vero cells are made from monkey kidneys. It's a cell line you can use for experiments. Gotcha.
Match out of all known species for SARS-2 receptor binding domain. And second, they talked about N-glycan manipulations, and I'll talk about this in detail. And SARS-2 has a missing N-glycan, and this feature actually greatly increases infectivity for human cell lines. So this is also quite interesting. And finally, furin cleavage sites. That's I think I already talked a lot about this, but it's just very, very weird that SARS-2 has that unique feature.
So I guess that it'll be interesting to just counter arguments to this. But so my understanding is you would say that the spike protein that did develop with the increased affinity for the ACE2 receptor wasn't predicted by computers, so it's unlikely that they would discover that in a lab. Although maybe for some reason this one they would. I'm just giving what the counter argument would be. That N-glycans tend to, I don't know what the counter argument is. This isn't related to the polybasic thing, I don't think. No. And then for the FCS, furin cleavage sites have developed not in SARS coronaviruses or whatever, or co-viruses, but they have developed in other related coronaviruses on their own. But I don't know.
But just to make sure, like, we're not cherry picking, we've put together this table of everything that DEFUSE talked about, and also
That's
whether
good.
we see evidence of that or not in SARS-2. So just to quickly sum up what DEFUSE was about, it was concerned with novel SARS-like viruses that pose a higher risk of spillover to humans. So they were going to screen spikes from a whole bunch of SARS-like viruses. And then if they saw good binding, then they would also verify this in vitro by slicing those spikes into chimeras. And if they saw again good results from chimeras, they would validate those results either in culture or maybe in live humanized mice. And of course, the goal was to see also the pathogenicity that these viruses pose to humans, and humanized mice are a good model for this. And
I wanted to ask a question about this slide as I was reviewing, and just kind of like a higher level question. A lot of the proposed work in DEFUSE, and much of it you've highlighted here, seems like it would leave vendor records, something where either they've purchased H2 mice or they talk about primer walking, they're going to purchase all those primers. There are all sorts of things that might give evidence to which of the DEFUSE activities actually happened, since it wasn't funded. Has anyone looked for that information? Would it all be Chinese vendors and extremely hard to come by? Would any of it be from Thermo, IDT, etc.? Can you speculate, comment?
Actually, like SAR said, questions will be at the end, and then like this kind of gets. First of all, I didn't plan on answering them now because that kind of takes away from the time, and I'm like borderline on 90 minutes. So maybe we can.
Okay, we can address it later, but we had planned. I was under the understanding we had planned to ask questions during presentations.
Well, yeah, that way I won't make it 90 minutes. Sorry.
We can save on the interruptions, I think.
Okay, we'll save. I'll save that till later then.
Okay. Yeah, I can.
That's a fair question, but that might not be exactly what he is studying, maybe. But hold on. It's time to eat fresh. Feels like the moderator questions are tailored to the how I guess capable they feel like the debater is of answering the questions.
And definitely address those questions later on. Okay, so like back to the slide. Yeah, it just summarizes that what the DEFUSE plan to do and how we might see some of these things manifest in SARS-2, and of course. Like DEFUSE was not like WIV didn't have to follow DEFUSE to the letter, because I mean DEFUSE was not gospel. They could do whatever research they wanted, and DEFUSE was essentially just the minimal, like an abstract of a research paper, and like it had one-line things about like we'll generate null Furin cleavage sites. But of course, once you actually dive into the research that you plan to do, it can be much greatly wider than what you originally planned or what you described in the research proposal. And of course, also in terms of funding, just because like DEFUSE wasn't funded, obviously WIV didn't need the funding to do the things that they were interested in. They had plenty of Chinese funding. They were doing this type of work anyways from Chinese funding for years. They had all of the viruses in their collection from decade plus of collection of sampling bats. And in terms of vendor records. Just like it's hard because that thought is already in my head. I'm thinking about it. Like I'm not sure what kind of vendor records they would need if they were synthesizing the backbones of the virus. They would probably do that in house. So
there's no way they do everything in house, right? I imagine there's places where you order these mice from, or other laboratories that you send stuff off to for testing. Like I think there's a big like verticality to a lot of medical research where there are different labs that do different things that I would be surprised if every single part of this is in-house, but I don't know 100%. I've known friends that work in labs before, but...
because they created backbones of the viruses in-house for the PhD thesis. Like they had two different backbones for WIV1, and so even if they used outside vendors, they probably would be Chinese vendors. So anyways, kind of going back to the DEFUSE proposal, as I said, there's some interesting aspects of DEFUSE that or interesting aspects of SARS-2 that sync well with DEFUSE. Of course, the primary one being or one of them being how well it binds to the human ACE2 receptor, which of course was the selection criteria for them to look at interesting viruses. And of course, several computer models and infectivity experiments have shown that the human receptor is like, if not the highest, among the top highest species in terms of binding affinity of the SARS-2 receptor binding domain. But one thing I want to point out is that just because I mean, that alone, the high affinity to the receptor, that probably would not be enough to spark a pandemic, because you need a furin cleavage site, essentially. Because we have experimental results in non-human primates that show that without a furin cleavage site, this virus remains an enteric gastrointestinal virus rather than a respiratory virus. Because basically we have, like in the wild, we have viruses like Banal52, Banal236, and they have the same receptor binding domain as SARS-2, but they don't have a furin cleavage site. And so there's been experiments by Marc Eloy's group at the Institut Pasteur, who actually, he's the one who discovered Banal52 and other Banal viruses in Laos. And so they've done experiments that showed that those viruses remain enteric, those viruses without a furin cleavage site remain enteric, while in non-human primates, in macaques, while SARS-2 actually becomes a respiratory virus in these animals.
When he says enteric, we saw that word earlier. What does enteric mean? Just internal, like it doesn't infect other people, or just gut? Okay, so related to the digestive system or your gut. Okay.
And this was actually corroborated by observations of guano collectors, people who collect bat guano in these caves where these viruses are actually present, like Banal52. And those guano collectors, they didn't have antibodies to like SARS-2 or Banal52, basically meaning that they were not infected by these viruses. And so I think this kind of syncs up well with the DEFUSE proposal, which was interested not just in the affinity of the receptor binding domain to the receptor, but also in the role of proteases, and especially like human-specific proteases, in like unlocking the barrier to zoonotic emergence, as like one other paper said, that protease cleavage unlocks the barrier to human emergence. I think in the 2015 paper with Ralph Barrick, where they engineered a null furin cleavage site in a MERS-like virus, that HKU4 that I mentioned before. So again, that's out of alignment. Yeah, besides the furin cleavage site, which I'll do an even deeper dive later, there's another unique feature of SARS-2 that corresponds to something that DEFUSE talked about. And this feature is a missing N-linked glycan at position N370. And this ablation of the glycan has not been observed in any natural relative of SARS-2 to date. And what makes this particularly noteworthy is that the ablation of this glycan significantly enhances the infectivity of SARS-2 in human lung cells. But at the same time, this mutation seems detrimental to ancestral bat viruses, because in the bat stomach, it is actually detrimental. And this is actually similar to furin cleavage sites, which are also potentially detrimental for enteric transmission, and so for which there's also selective pressure against in bats. And so DEFUSE proposal's interest in N-linked glycans, I think, well, it can be traced to the observations about SARS-1 that it lost 2 glycans in civet strains, and they speculated that this potentially helped it jump into civets, and then of course from civets it jumped into humans, and so they were interested in a role that glycans can play in like host switching mechanisms. And so this is the direct quote from the DEFUSE proposal. Basically it's talking about like what I just said, that the ablation of these 2 N-linked glycans played some sort of role in the virus's first jump into civets or raccoon dogs or both. Well, civets are the strains that we actually have in which those glycans are missing. At the same time, the DEFUSE authors now knew that in the original bat strains, the glycans were still present, so they went missing somehow. And
does this guy's argument seem more substantive to you? It feels like it isn't. Isn't it weird again? Well, he seems to understand the underlying material, which is good. But apparently this guy's like a PhD. Fuck, I don't remember what it was. It wasn't like a virologist, but it was something peripherally related, something in biology. So he's very familiar with underlying material. These are at least real arguments. I understand how these link. The last guy was like a meme, but these are real arguments at least. I can understand. They at least deductively are true. Like all of the arguments he's making are valid. So the question is going to be the soundness of the premises. Are the statements that he's making true? Like in all cases or in a way that's relevant to this. But these arguments are way more sophisticated than the last guy, for sure.
Then they infected civets, but then they reappeared during the human transmission. And so basically the DEFUSE authors were wondering what kind of role this ablation of these glycans might have played in helping these viruses jump into civets. And so they're citing this 2007 paper, the DEFUSE proposal, basically in connection to these two N-linked glycans. And they're showing that basically these 2 glycans at positions 227 and 699 are missing in earliest civet strains like this one and this one, but then they gradually reacquire them and preserve them in human strains. And so this is one of the motivations they list in the DEFUSE proposal for why they wanted to look at N-linked glycans, and another motivation was
But the scientists did diffuse because they predicted such a virus, so why wouldn't you expect it for it to happen naturally? We're in a deeper level of the argument than just talking about the virus. We're talking about specific portions now of the RNA and the proteins and stuff that are being expressed by it, and how likely it is for them to show up in certain populations. So he's saying that there's some adaptation having to do with N-glycan, and that adaptation is something that would be beneficial while infecting humans. So you might see an evolution towards the development of these N-glycans inside human hosts. However, it would be predicted that these would actually cause a decrease in infectivity in bat hosts. So if your guess is that this was a zoonotic transmission, there was a crossover event from animals to humans, he's saying that the presence of these N-glycans is more accurately predicted when human hosts have been transmitting it or when it's been engineered, and it would be like not predicted in at least bat populations. Now, I don't know if the fixation on bats here matters, because you could maybe make an argument that it came from some other animal, but that's what this is, yeah. That's what his argument is here.
The role that the glycans play in this DC sign binding, this binding to alternative receptors. And also, of course, as I mentioned, they said that while those glycans were missing in civet strains, they weren't missing in progenitor bat strains, so they had this kind of flip-flopping role between bats, civets, and humans, and so they wanted to investigate that. But in terms of the DC sign binding connection, they then cited a 2006 paper that talked about this particular glycan that they identified in SARS-1 that they think played a role in DC sign binding. And it's interesting that the very same glycan that they mentioned in that paper is the same glycan in SARS-2. Like N370 in SARS-2 is the same as N357 in SARS-1 that was linked to this DC sign binding. And so, of course, N370 is not the same glycan as the civet glycan, but it's the same glycan as this DC sign glycan. And that section in DEFUSE, it talked about glycans in connection to DC sign binding, which is a receptor in macrophages. And basically the idea is that maybe if you ablate this glycan, you can increase the virus's ability to infect other tissues. Maybe you have a reduced titer in macrophages, but maybe it now has the ability to infect other tissues because macrophages play a role in immune response to it. But I won't speculate like exactly why they were interested in this. And also, like, I'm not saying DEFUSE is the exact blueprint that WIV would follow to the letter. Again, like they didn't have to ablate the very same glycans, but it's just showing that they were looking at glycans. And if someone later, inspired by DEFUSE or just in general interested in the role that glycans play in host switching, and someone had this panel of various viruses that they wanted to see the role of glycans, they could have been ablating them one by one and seeing how that affects infectivity and maybe noticing that increases infectivity in human cells, human lung cells. And then they would have done, for example, in the SARS-2 progenitor, this very same experiment that would have resulted in the loss of this glycan.
Why are the debaters different from yesterday? Well, this guy's the same because he's the one doing the debate challenge. Yuri has been brought in to do this part of the debate, I guess because he has a specialty in this area. I don't really know why it's not the other guy doing all of it, but the other guy very clearly doesn't have he's like the other guy is what people accuse me of being. It seems like he has a very loose grasp on argumentation, and then he thinks that could carry him through the whole process without knowing any of the underlying facts, basically. I thought they said SAR was busy. I don't believe that.
And yeah, basically it's the importance of this glycan in SARS-2 is that it greatly increases infectivity to human cells. And this is a quote from a study they did basically showing that when you have this mutation, it increases infectivity in human cells by like 60 times. Basically, if you restore the glycan, the wild type mutant, it has a 60-fold lower infectivity than the current SARS-2 that has the missing glycan. And just to provide some comparison, like
okay, well, fuck me. This guy said, Corey Campbell says, FYI, the Yuri Dagin guy does not have a PhD. They have a bachelor's in comp sci, according to their LinkedIn. They just happen to work in biotech as an entrepreneur. They're not speaking like a specialist. They're actually saying some silly things. Okay, well, if he's saying silly things, I can't identify them, so we'll wait until our god responds.
The G614 gene mutation, the early mutation that is known to have increased...
Regardless of if he's saying silly things or not, though, like, he still has a much stronger command of the material than the guy from the previous guy who had zero understanding of literally anything, like data or underlying, biology stuff.
Comparison, like the D614 gene mutation, the early mutation that is known to have increased SARS-2 infectivity, it only boosted it by like 3 times. So that's a huge... Then this glycan plays a huge role in increasing infectivity in human cells. And so I think, again, in light of the DEFUSE proposals focused on N-link glycans and their potential role in host jumping, and as we now know, infectivity, I think it's very, very interesting to see that SARS-2 is missing one of the glycans that potentially DEFUSE might have been interested in. And it just keeps getting out of alignment. Okay, yeah, and as I said, while the presence of this mutation in SARS-2 significantly increases its infectivity to human airway cells, in bats, this mutation is actually detrimental. And so it's not likely to have arisen there. And this is because these viruses predominantly rely on like oral fecal transmission route, and this mutation, is, as this quote shows, is detrimental to oral fecal transmission, basically because of the like low pH in bat stomach. So, well, I won't get into the details, but basically they did a study that they restored this mutant and checked how that, like what the implications were on the virus, and they showed that the mutants lost almost all of the infectivity under the conditions of a bat stomach, while the wild-type viruses, the viruses that actually had the glycan, they retained a significant portion of the infectivity. And so basically this tells us that while these the ablation of these glycans is an advantage for human infection, like in human airway cells, it's actually detrimental to the virus's ability to transmit via the oral fecal route among bats. So this kind of, well, I think it maybe not rules out, but like greatly decreases the probability of this mutation arising in bats. Okay, so like setting DEFUSE aside, even if we didn't know about DEFUSE, like we didn't know about DEFUSE until like summer of 2021. So, but even then, when we see some features of the SARS-2 genome, some of them are like very, very surprising and like not in line with evolutionary expectations. As Christian Andersen said early on, he was the original lab leaker. Yeah, one of course, yeah, the Furin cleavage site is probably the most distinguishing such feature. And I mean, of course, sometimes Furin cleavage sites do occur in coronaviruses, like as a family.
Okay.
In SARS-like coronaviruses, SARS-2 is unique among like 1,500 known SARS-like coronaviruses in having this Furin cleavage site. So this is just so much like unexpected to see a virus from this family with a Furin cleavage site, because also, as I said, there's very likely selective pressure against it, because it's actually detrimental for the gastrointestinal tropism of these viruses. Because it shifts, like if you add a Furin cleavage site, as Mark Eloise has shown experimentally, that shifts the tropism from enteric to respiratory tropism, and of course, yeah, bats prefer to have enteric tropism. And just not only that, but also the way in which the Furin cleavage site arose is also very unexpected, that it's not like a single point mutation, but it's, which would have been enough. Like there's some viruses which are only like one nucleotide mutation away from having a Furin cleavage site arise. But this Furin cleavage site was introduced by like a 12 nucleotide insertion at like this very same spot that created the Furin cleavage site, which is very suspicious. At the same time, like given that the pandemic, I mean, this was originally a bat virus, so for us for this bat virus to...
Oh, I just understood that. For some reason, I was thinking nucleotide insertion was like inserting the RNA into the cell, but that doesn't make sense. 12 nucleotides wouldn't be enough to encode for anything. It's saying that FCS was encoded for with a 12 nucleotide sequence, so it's not like some random like swap or some random mutation in the RNA strand produced this. It took 12 nucleotides were necessary to be inserted to have this FCS appear. So this likely came from some other thing, or was... Can RNA stuff like be recombinant in the wild? Can like a ribosome like pick up 2 RNAs and try to print shit off of it, or... So it would, but I'm guessing the structure would have had to emerge from somewhere else or something like that, or I guess not necessarily. It could just randomly, because that chain of amino acids could just be functionless or not do much. But then when enough of them come together, they have some emerging phenotype. Maybe, I guess.
And kind of start infecting humans. It's not unreasonable to anticipate some unique feature that will allow us to do this. So, by itself, the Furin cleavage site, while it's very kind of unusual to see, from a probabilistic assessment, I think, yeah, we're not giving it a very high weight. But the particulars in which it came to be, they are the more suspicious. As I said, the clean insertion, the 12 nucleotide insertion in the spot that created it, as well as also the choice of the codons, the CGG codons for arginine, the CGG doublet. It's a very unusual codon choice for coronaviruses as a whole, and but wouldn't be unusual for genetic engineers. And not only it's unusual, we've looked at for whether there are other SARS-like viruses that have a CGG arginine doublet. And in response to like Tyshkovsky and Punchin, we analyzed 255 existing coronaviruses, SARS-like coronaviruses, and we haven't found a single one that used anywhere in the genome a CGG arginine doublet. And I mean, it's just very, very unusual to see, and also to see an insertion in the...
It's funny. It seems like this guy has a stronger command of the molecular biology part, but I wish that he would dive in a bit more to the probability part, because the probability matters here, and the scale is incredibly important. When he says very unlikely, and we're talking about like infections and pandemics, like there's a big difference between something being very unlikely at like 0.1% versus very unlikely at like 0.1 e to the negative 27 or whatever. Like there's a huge difference in likeliness there. I wish that he would, as a part of his argument, I wish he would kind of like peg these on some kind of probability so that you could get an understanding of it, I guess. Or at least I don't understand, because he's like, oh, it's very unlikely for this to happen. Well, sure, but I mean, like, I'm sure, how many animals are spreading these viruses back and forth over and over and over again? What do you mean by very unlikely? Like, it could take, like, very unlikely as in, it's unlikely that you would guess a password on a computer and it would take another computer 1 week to guess the password, or it would take the heat death of the universe to guess the password. Both of those are very unlikely to be guessed by a human, but there's a huge difference in magnitude there between the unlikeliness,
The genome in general, but also that precise spot of the genome in which it's inserted. If you look at the alignment of SARS-2 relative to its closest ancestor or closest relative, Banal52, you'll see just like how close they are in the latter part of the genome, basically how rare even mutations are. And so it's unprecedented to have such a major like insertion, a pretty long insertion that has such an important functional consequences to occur in this spot of the genome. So basically, this is an alignment of the entire latter third of the virus, of the genome, and it's just trying to show like just how rare even non-synonymous substitutions are in this part of the genome. And basically, yeah, it's pretty much, the insertion is unique in this part of the genome, this long of an insertion. And that alone is surprising, but...
Do these virus cleavages have OnlyFans? These viruses are like 4 or 5 years old, you sick fuck.
In this part of the genome, this long of an insertion. And that alone is surprising, but of course the functional significance of this insertion being at the very precise spot of the cleavage site is even more surprising. So, I mean, these are just looking at the alignment, and basically you have some hotspots of single nucleotide mutations. Some of them are synonymous, some of them actually change the amino acid, but there's just not too many places where you see such mutations. And then you kind of see this horn of the unicorn, to borrow from Alina, the analogy, that just sticks out. And it's not, again, it's not just any odd place in the genome where it occurs, but it occurs precisely at the cleavage junction of the S1 and S2 subunits of the spike protein.
So like my question here would be, so I see we've highlighted SNPs, and then we've highlighted here the sequence of 12 nucleotides that are responsible for the FCS be created. But the way that this looks to me is the intuitive feeling here is, oh, okay, wow, all 12 of these changed at the exact same time to cause this FCS to emerge. This is a very clean insertion. It seems highly unlikely. However, one, I don't know how likely that change would be, but also two, is it not possible that all of these encode for something that's like non-functional or junk, and then it took one more mutation for it to finally become an FCS? So maybe there were prior, ancestors or generations or whatever that had almost all of these, but they were missing just a key amino acid flip or mutation, and then it became an FCS? I don't know the answer is to that, because if that is the case, then this is, I would say arguably this is a bit misleading, right? Because couldn't this theoretically be a ton of non-functional genetic drift, basically, and then one mutation caused the emergence of this functional FCS? And then if you go back and go, oh my God, it's crazy how that was so, like if you look at the evolution of the human body, our eyes didn't just emerge. And like, oh my God, what are the chances that we have DNA that encodes for an eye? It's like, it was a whole bunch of small changes, some non-functional, some functional, that built up to the eye we have today. I don't know.
And in the vicinity of this insertion, there's just, again, very few mutations and very few non-synonymous mutations, and yet it kind of sticks out as being in this very kind of low mutational area, and yet it created this Furin cleavage site. And I mean, the later part of the genome, sometimes you have these mutational hotspots, but that was not like that general vicinity of the spike protein was not a mutational hotspot. And I'm out of alignment again. So yeah, it's just very odd to see an insertion in general, because insertions in general are pretty rare beasts. And this slide just try to show how rare they are, because we have a very good now picture of the mutational propensity of SARS-CoV-2, because we now have like 10 million SARS-2 genomes sequenced throughout the pandemic, and after like it infected close to 1 billion people probably by now. I mean, this is like, anyways. So we have this distribution of the mutations, and insertions are by far the rarest mutation. This green line in this bar chart are insertions, and this is the spike, and you see that it's like very few, like insertions represent very small percentage of mutations in the spike. And moreover, most insertions are actually short, and 12 nucleotide long insertions are also rare. And of course, insertions really have to be in a multiple of 3 to preserve the reading frame, because codon is 3 nucleotides long. And so we have like all throughout 10 million known SARS-2 genomes, we have like 35 12 nucleotide long insertions after like years of evolution in probably 1 billion people. And yet, of course, most insertions don't even happen in a spike.
Most insertions don't have, yeah, but I'm curious, I just don't have the background. These are just questions. Maybe he'll ask, or I wish somebody would ask, like, I imagine that the natural selection, the selective pressure on the evolution of the virus is going to change significantly once it's entered and is wildly spreading through human populations. That would be my guess, that if you look at the way that the virus has mutated, if it is of zoonotic origin, the way that it mutated prior in animals might be quite different than how it mutates in humans, because different selective pressures exist, right? Maybe, I don't know. Maybe that's not the case. Maybe they would say, actually, no, because of the way because of what we're encoding for in this virus, and because of the similarity in the types of receptors and everything else that it targets, we wouldn't expect to see a significantly different pressure. Although, I mean, with the N-glycans thing, apparently there are selective pressures that are different between, one species and another. So I don't know the answer to that. I wish he would speak more about that. But again, maybe he'll get into it. And he can't do everything. He's got a 90-minute presentation. So
it's shown here. So not to belabor the point, but it's a very, very unusual insertion to see in a SARS-like virus. And as I said, another interesting aspect of it is that the insertion has arginines coded by the CGG codon, and it's a very rare codon for SARS-like viruses, and in general in viruses they don't like this codon. And as I said, we looked at not just the codon, but at the doublet, the CGG doublet, in our paper in response to Tyszkowski and Panchin, and we haven't seen this doublet in any of the other SARS-like virus that we looked at. And basically 2.6% is the frequency of arginines being used by, being coded by CGG in the SARS-2 genome, and there was some questioning whether is it exactly 2.6 or maybe it's a little higher, and we found another source for our numbers, and basically, this is a very nice table that shows all of the arginines in each gene of the coronavirus, and then we can just count up which ones are coded by CGG, of course, omitting the insertion. And basically, without the insertion, only 2.6% of arginines in SARS-2 were coded by CGG. And moreover, in the spike gene, only the insertion has CGG. So in the spike, it's even less likely for us to expect to see a CGG codon to be used, let alone 2. And of course, you might ask, why would any lab...
Okay, I'm sorry, I need to understand this better. Okay. Arginine is it would be like some amino acid plus the 5-phosphate things plus whatever. This is like our, it's not the, what do you call it? What's the name for this? It's an amino acid plus the other thing. So it's like an amino acid. We can just say amino acid, I guess, right? Or the other name that they had for this was residue. Is that what they would call it? Just, okay, we can say it's an amino acid. Okay. Wait, arginine doesn't have phosphates. Oh, okay. So is this saying, when it's saying that these, this codon encoding for this amino acid is rare, is the implication, when we look at, okay, wait, when we look at CGG, CGG, each of these are nucleotides? Yes, okay. Is it saying that you can encode this amino acid arginine, that you can encode this with a different sequence of nucleotides? Is that okay? And they're saying that for arginine to show up as an amino acid, this particular sequence of nucleotides is not the common one that you expect to see arise in nature. Again, I wish we could give percentages. Maybe it's written here, but okay. The last letter can be different and still form an arginine. Okay. DNA and RNA codon tables. Holy shit, fuck that. What the fuck? What, so this means A, G, A, and AGG both make arginine? Is that matching with what he's saying? Why does he have CGG? He has C. Oh wait, there's a different arginine. What the fuck? Oh no, wait, that's the same thing. This is the exact same amino acid, correct? Even though a different set of nucleotides could give rise to it. Okay. It's still functionally the exact same thing. Okay. Gotcha, okay. Look up the circle one, it's easier. Okay, this is CGG, CGG. So CG, no, wait, they're not that one. Wait. Oh no, wait, you go out. You go out. I see. You started in the middle. Okay. Fuck me. CGG. Okay. Oh, the last amino acid really doesn't fucking matter at all. No, I'm sorry. The last nucleotide doesn't matter at all for the creation of the amino acid. Okay. Rare codons refer to the number of tRNAs that can recognize a specific codon. It has nothing to do per se with the codon itself, but the number of tRNAs that can recognize it. tRNA, is that transcription? I don't know. Maybe. Okay.
Or WIV or whoever use CGG codons if they were to insert a CGG, arginine furin cleavage site. And I mean, first of all, we don't know the exact motivation of somebody, but there's some plausible explanation. The first one being just that it's the kind of the most top of mind codon for human genetic engineer, as this table shows from the Moderna and Pfizer vaccines. When they were codon optimizing their spikes for the vaccine, they recoded either almost all of the arginines to CGG, or the arginine starting with a C to CGG. And so it's just, I think it's a natural codon for a human genetic engineer to use, just because I think it's in the human genome, CGG is actually either the most frequently used codon or like at the top of the codons being used for arginine. Also,
this goes into it's separate from this argument, and I don't know which lab leak theory that he's arguing for. I don't know if he's arguing actually for specifically for any of them. He might just be saying that the presence of this codon to encode for arginine is weird. But if you were arguing for any sort of bioweapon, if that was what you were arguing for, I imagine that unless this codon has other properties that make it more resistant to mutation or something like that, I don't know, maybe they do. It seems unlikely that you would choose like the rarest codons to create your bioweapon with, because it would be so obviously identifiable, right? Unless there's some engineering advantage to inserting these types of things, to doing CGG as opposed to, CGC or whatever might be more common. But I mean, he could be arguing just for a non-malicious lab leak and not like a bioweapon creation as well. That could be the case too. So, I mean, again, I don't know. The bioweapon lab leak theory is the most aggressive and kind of conspiratorial one you could argue for. So,
this is my favorite part. CGG creates a new restriction enzyme site for this FAO1 restriction enzyme that could be useful in tracking the newly created furin cleavage site, because as we now know, these furin cleavage sites can sometimes get deleted if you passage it in like the wrong cell culture that doesn't express furin or has very low furin expression. And so if you're tracking the evolution of a virus, if you're a genetic engineer, it's nice to have this easily accessible tracking beacon in the form of a restriction enzyme site that you can quickly do a digest and like run a gel and see if your furin cleavage site is still present in your viral colony or if it's a mutated way. And when I pointed out, Peter mentioned that he hasn't observed anyone in virology using FAO1 before, but it actually has been used. And so here are like examples of a couple of papers, and one of them actually used FAO1 for this RFLIP analysis that I mentioned or hypothesized could be useful for SARS-2. And RFLIP stands for restriction fragment length polymorphism, and it can be used to identify the presence or absence of specific restriction enzyme sites, which can then be used to like quick screening of deletions, for example, of the furin cleavage sites. And
yeah, so like Wang Sima says, no one uses a restriction site as a tracking beacon for that. It's like, I wouldn't have no idea. I don't work in a lab. I don't know how people track any changes or how easy or difficult it would be to analyze any mutations from generation. I have no idea.
Yeah, like I said, because furin cleavage sites, as we now know, are prone to deletions in SARS-2. Okay, now let me address another aspect of the furin cleavage site, the leading protein. Basically, some say RRA would have been enough to create a functional furin cleavage site, so why does SARS-2 have PRRA as its insertion? But even some of the proponents of the zoonotic hypothesis, they've suggested in their studies that RRAR by itself, without the proline, might not be actually all that well cleaved. But if you introduce a proline, it creates a turn in the protein, which actually could expose the S1, S2 junction of the protein, that loop that contains the null furin cleavage site, and that can actually enhance its cleavage. And I mean, yeah, that's just kind of one idea, but just to kind of like dismiss the whole lab leak origin hypothesis by saying like nobody would insert a proline, I think it's not a good argument in general. I mean, genetic engineers could have their like many reasons for why they would want to use to include a proline in their like leading proline in their insertion in their furin cleavage site, because for example, MERS, the MERS coronavirus has a leading proline in its furin cleavage site. Some feline coronaviruses do, and WIV was like they studied MERS a lot. In fact, they engineered a novel furin cleavage site in a MERS-like virus, showing how important the furin cleavage site was for unlocking human tropism. They also studied feline coronaviruses, and at the same time, there's some naturally occurring coronaviruses, like close to SARS-2, that have very close resemblance to the SARS-2 furin cleavage site. And for example, the viruses like RM102 and Banal 116, they have this PAAR sequence at the S1, S2 junction. Of course, PAAR is not cleaved by furin, but it's because you need arginines. And so basically, if you insert RR in there, and if you turn PAAR into PRRAR, that actually gets cleaved into furin by furin. And so they could have been modeling the evolution of what would happen if this PAAR evolves into some version of a furin cleavage site, and they could have had several candidates for cleavage sites from like the most minimally cleavable to like very highly cleavable. They could have a panel of various furin cleavage sites they wanted to evaluate. And it's actually very interesting.
45 minutes.
Because, if you, it's interesting because if you look at the actual nucleotides of the PAR fragment in these natural viruses, they use the same codons, like CCT for proline and GCA for alanine, as is used by the PRA insertion. And so it's like if someone took the PA fragment and inserted from these natural viruses and just inserted 2 arginines in between. So, I mean, of course, you could say it's just could also happen just by chance, but it's just a very interesting coincidence in a series of a whole bunch of very interesting coincidences. And so, basically, yeah, this is what could have happened is a genetic engineer would find like a RM102-like virus, see PAA fragment in there, and decide to model like the evolution of that into PRRAR, furin cleavage site. And so I think it's quite plausible that somebody at the Wuhan Institute of Virology or any other lab in Wuhan looking to, inspired by the DEFUSE proposal, looking to carry out this kind of research into modeling the spillover potential of these viruses could end up with a furin cleavage site with the leading proline. Okay, so in summary, the furin cleavage site.
Wait, so real quick.
SARS-2 stands.
When we say
in summary,
what is a leading proline again? What is a proline? For the cleavage site, it's just something that causes it to morph or twist or turn more once it's activated before the cleavage site. Okay, so ideally, and his theory was that like this could help expose it more to whatever's cleaving it, the furin. Okay.
Modeling the spillover proline. Okay, so in summary, the furin cleavage site in SARS-2 stands out not just because of its existence, but also because of the way that it came into existence. And of course, while it's not impossible for a virus to evolve a furin cleavage site naturally, the conditions and characteristics surrounding the SARS-2 furin cleavage site, I think they make it a unique case, especially in light of selective pressure against an FCS in bat SARS-like coronaviruses. And well, in general, it just kind of very unusual to see such a novel feature arise, like a novel trait which was not seen before in any of the member of the family, arise in a virus that jumped species. And also, I think we would expect such a feature to arise via single nucleotide mutations or just mutations rather than an insertion, because as I said, some of these natural viruses are just like 1 or 2 nucleotide mutations away from.
He's frozen for me.
Like that way.
I can see you.
Whoa,
Eric's there.
What?
I just lost Jerry.
I think this shows that there's actually selective pressure against furin.
I paused his time. I'm gonna.
And also insertions.
Oh, no.
This is like the part of the Tour de France when the rider falls down and I have to go back and
come
help.
from the same virus. This is another aspect that, we still don't know where the insertion came from. Like, what's the donor sequence for the insertion? And usually the donors come from the same.
Hang on.
Sorry?
You cut out for about 15 seconds, so I'm not sure what we missed.
More than 15 seconds. None of us could hear you.
Yeah.
Go
I
back to the
paused your
beginning.
time. I don't know how far back you want to go. Maybe 30
Okay.
seconds.
The start of this slide is probably good.
Okay, start of the slide. Yeah. Basically, I was saying that it's interesting that it's not just the way that the kind of key observation is not just that we have this furin cleavage site in SARS-2, but also the way it came into existence in SARS-2 that is very suspicious. And basically, also I think I was saying that the way that it evolved using an insertion is very suspicious or very unusual, because usually like new traits arise via mutations, and this trait could have easily arisen via single point mutations, because viruses like RM102 or Banal 116, they're just one mutation away from a furin cleavage site arising, and yet we see this arise via this very uncharacteristic 12 nucleotide insertion. And so insertions, they can happen, but usually they're smaller insertions, and usually we actually know where they came from. Like the donor sequence of the insertion, and usually the donor sequence is either the same virus, same genome, because it usually happens during recombination events when the virus replicates. So either the same virus or some sort of like virus in the family that infects the same cell along with this virus.
Okay. the FCS, the furin cleavage site, and SARS-CoV-2 is encoded by CCU, CGG, CGG, GCA. So those are 4 codons of nucleotides, and they result in PRRA, which are 4 amino acids, and the P in the beginning, does that P stand for a thing? Is that a thing? Pro? Is it this? Oh, it's a thing. It's an amino acid. Proline is an amino acid. I was looking at it as like proline. Okay, proline is an amino acid. This leading amino acid in a protein structure is known for causing twists or turns, and that twist or turn is exposing the RRA, that furin cleavage site, to make it more likely that it will be cleaved by furin. That's so when we say leading proline, we're saying that of the 4 codons, the proline amino acid at the front exposes the FCS. Yes, okay, that's what I mean, right? Okay. Proline is a molecularly unique amino acid that, because of its structure, is much more rigid than the other amino acids. Because of this, it doesn't fit into standard protein shapes, such as alpha helices, and often causes the end of those shapes. Okay. Okay. And then, fuck, I keep forgetting this. Furin, the furin cleavage stuff doesn't happen until after it's binded to the ACE2. Furin cleavage stuff has nothing to do with the ACE2 stuff. Oh, no, it does. No, it doesn't. ACE2 is it binding to the cell, and then the furin cleavage. Furin is the enzyme that comes out and cuts it twice, which allows for the stuff to get come into the cell. No? Wait. The MIW says no. And then he said based and true. Okay, well, all right, whatever. Or close enough, I'll assume.
But in this case, we still don't know where the insertion came from. Like, what's the donor sequence with these 12 nucleotides that, gave SARS-2 the nucleotides for the furin cleavage site. And so, and finally, the codons, of course, yeah, they're also suspicious because they're not used. First of all, yeah, viruses like SARS-like viruses don't like these kind of codons, and we don't see them in the spike all that often. So, in light of all of these things, I think it's just SARS-2 is again inconsistent with evolutionary expectations.
Furin cuts the virus after it's made. Oh wait, yeah, the MIW keeps saying that. Nobody confirmed that. Where's my other guy in chat? Wang Sima contention. Is that true? Because in Pituku, they keep disagreeing on that. So the MIW is saying that furin only starts cleaving stuff once the virus is created. Is that true, or? Wait, that doesn't make sense. How could it have 2 different cleavage sites then? It would just cut off the first one and then you have a separate virus, no? Fuck, okay. And then the MIW stops. He never elaborates. Okay, I'm just going to keep assuming it's to admit entry into the cell.
Christian Anderson pointed out. Okay, let's move on to our third point. It's just it's intriguing that SARS-1 exhibited very low genetic variability pretty much from the start. And so typically when zoonosis happens, the virus actually has a very high mutational rate because it's trying to adapt to a new host. It's really kind of experimenting and trying to see, I won't use like anthropic language, but basically with SARS-1, for example, we saw a very high mutational rate and it made multiple jumps into.
The furin cleavage site and the SARS-CoV-2 spike proteins require transmission parents. Okay, SARS-CoV-2 entry requires sequential cleavage of the spike glycoprotein at the S1, S2, and the S2 cleavage sites to mediate membrane fusion. SARS-CoV-2 has a polybasic insertion at the S1, S2 cleavage site that can be cleaved by furin using lentiviral pseudotypes. Oh, we know what these guys are. In a cell culture adapted SARS-CoV-2 virus with an S1S2 deletion, we show that the polybasic insertion endows SARS-CoV-2 with a selective advantage in lung cells and primary human area epithelial cells, but impairs replication in VRE6, a cell line used for passaging SARS-CoV-2 using engineered spike variants and live. This isn't helping us at all. Coronaviruses enter host cells via their spike glycoprotein, which is synthesized as an inactive precursor that must be cleaved to mediate membrane fusion. Depending on the sequence of spike at the S1-S2 junction, the cleavage can occur 1, during trafficking in the producer cell by host furin-like enzymes, or 2, by serine proteases such as the transmembrane protease serine 2 at the cell surface during attachment, or 3, the cathepsin proteases in the late endosome/endolysosome. It happens before the virus exists in the cell. He's saying that, the guy in chat keeps saying that cleavage is what happens when the ribosome is printing the RNA. That's what it sounds like you're saying. Look at the picture in this paper. I was not chatting, trying to look for the paper. Which picture? Give the figure number. Figure 2 has a good picture. Which picture? Oh, figure 2. Okay, figure 1. Oh boy. Hold on, we're about to go. Okay, we're going to fully understand these two pictures. Good luck, okay? Okay, the nucleus here is the nucleus of the cell. What is ER? Endoplasmic reticulum? I don't even know why the fuck I remember that. I don't even know what the fuck that is. I don't know. I feel like I memorized this for like fucking biology in 5th grade. Is a part of a transportation system in the eukaryotic cell and has many important functions such as protein folding. The word endoplasmic means within the cytoplasm, and reticulum is Latin for little net. It is a type of organelle made up of 2 subunits, rough endoplasmic reticulum and smooth endoplasmic reticulum. He says again, eukaryotic? Okay.
Ecosystems, including relatively novel eukaryotic cells.
We explain the differences between prokaryotic cells and eukaryotic cells.
Okay. So, nucleus, endoplasmic reticulum, replication organelle. What organelle is it? What does that mean? Transfer of viral structure proteins and genomic RNA. ERGIC. What is ERGIC? Endoplasmic reticulum Golgi or Golgi? Golgi intermediate compartment involved in protein transport between the endoplasmic reticulum and the Golgi or Golgi.
Organelles that would be found exclusively in eukaryotes, such as the nucleus, endoplasmic reticulum, Golgi apparatus.
Golgi apparatus.
Static hold for 60 seconds to override that Golgi tendon.
Mm-hmm.
Okay. Plasma membrane, is this just the thing that gets you into the cell? Cytoplasm is all the gunk in here. Okay, this is our SARS-CoV-2 virus. Okay, infection by a coronavirus induces in the paranuclear area. Paranuclear area, does that mean area around the nucleus? Infection by coronavirus induces in the paranuclear area the formation of new membranous
which is the big fluid-filled system of wavy wormholes, and the membranous labyrinth, a continuous series of sacs and ducts.
Thanks.
Germ granules are indeed membraneless granules. And then I will...
Thanks.
Kidney, like IgA nephropathy or membranous nephropathy.
Thanks. Thanks. Infection by coronavirus induces in the... Fuck it, what is paranuclear? Tell me. How do I say it?
Perinuclear region.
Peri.
And the peripheral region.
Peri.
The gradient is destroyed because...
Shut up, loser.
And in these regions that we call perinuclear regions, which is where the Golgi resides.
Immediately start. Okay, so it's not. It's just a place where things happen. Okay. Okay. Infection by a coronavirus induces in the paranuclear area the formation of new membranous. I don't remember what it was. Membranous structures of various sizes and shapes, which as a whole are referred to as replication organelles. Okay. These structures, okay, these are new things being made. Crazy, okay. These structures, observed by electron microscopy in cells infected with mouse hepatitis virus. Is a hepatitis virus also a type of coronavirus? Nope. Okay, they're just adding it in here because why the fuck not? Okay. These structures, observed by electron microscopy in cells infected with mouse hepatitis virus, severe acute respiratory syndrome coronavirus, or MERS, Middle Eastern Respiratory Syndrome coronavirus, and typically surrounded by double membranes, likely originate from the endoplasmic reticulum and house viral replication complexes, sequestering them from cellular innate immune molecules. Okay. I didn't know any of this. I thought that the RNA just how the fuck does it even replicate itself with... Oh no, we'll keep reading, I guess. I don't know. All right, let's see. Viral structural proteins and genomic RNA synthesized at the replication site are then translocated through an unknown mechanism to the Golgi intermediate compartment where virus assembly and budding occur. Okay. Viral structural proteins and genomic RNA synthesized at the replication site are then translocated through... Oh. Okay, I guess that actually kind of makes sense. It would have to, because otherwise, if the virus were to insert itself and then the RNA was processed by the ribosome, you would just get one fucking virus, right? You would have to produce even more RNA inside of the cell, which is what these replication organelles do, I imagine, right? That's what's... Okay. This animation shows this pretty well. Okay, maybe we'll look at that after. Viral structural proteins and genomic RNA synthesized the relationship and then translocated through an unknown mechanism to the Golgi intermediate compartment here, where virus assembly and budding occur. Only 4 viral proteins, the spike, envelope, membrane, and nucleocapsid proteins, are incorporated into the virion. And the virion is the name given for the entire virus, including like the envelope and everything else on the outside, right? There's an enzyme encoded by the virus, RNA-dependent RNA polymerase. It does, yeah, okay, the virus multiplication, okay. While the N protein bound to the viral genomic RNA is packed inside the virion, the structural proteins, S, E, and M, S is the we knew these. S is the spike, obviously. Was M just membrane, and then E is entry or something to get in for some to get in? I don't remember. Fuck, I don't remember. Fuck it, who cares? No, I do care. Fuck me. Oh, envelope. No, I don't care. No, fuck it. Okay, that's fine. Oh, it's literally right here. Yeah, just kidding. Jokes, trolls, trolling. Good job, you paid attention. You passed the test, Jack Darko. And Disco the Q. The S protein assembled as a trimer, a polymer comprising 3 monomer units, a molecule that can be bonded to other identical molecules to form a polymer.
Monomer.
Polymer, monomer, not polymer. Polymer, monomer.
Trimer.
No, sorry. A monomer, trimer. Okay. The S protein, the spike protein, assembled as a trimer, giving the appearance of a crown or corona, mediates major entry steps, including receptor binding and membrane fusion during biosynthesis and maturation in the infected cell. The S protein is cleaved by furin or furin-like proprotein convertase in the Golgi apparatus in the S1 and S2 subunits, which remain associated. Okay. This makes, okay. So this cleaving shit does happen inside the cell. Okay. The S protein is cleaved by furin or furin-like proprotein convertase and the Golgi apparatus into the S1 and S2 subunits, which remain associated, as in still connected, right? The S protein on the virus, therefore, consists of 2 non-covalently associated subunits with different functions. In the new target cell, the S1 subunit binds the receptor, and the S2 subunit anchors the S protein to the virion membrane and then mediates membrane fusion. The E and M proteins contribute to virus assembly and budding through the interactions with other viral proteins. Assembled viruses bud into the ERGIC lumen and reach the plasma membrane via the secretory pathway, where they are released into the extracellular space after the virus-containing vesicles fuse with the plasma membrane FP fusion peptide. Okay. Jesus fucking Christ. The S1 subunit binds the receptor. What does that mean when it says binds the receptor? And the S2 subunit anchors the S protein to the virion membrane. So the S2 subunit keeps it anchored into M, and the S1 subunit binds the receptor. It's just part of the receptor that binds it to the ACE2 thing, right? Okay. So then when we say cleavage, yeah, okay. So then when we say cleavage, okay, we've almost got it. During biosynthesis and maturation in the infected cell. So this means that when the replication organelle sends its goo to the Golgi, I guess, the endoplasmic reticulum Golgi IC, while it's in there, are ribosomes present there? So then once it starts printing, the furin comes by, the S protein is cleaved. Oh, yeah, in the Golgi apparatus. Okay. It just needs one furin cleavage site as well, right? FCS, one furin cleavage site. It's cleaved into the S1 and S2 subunits, which remain associated. Okay. Jesus. Okay. I don't know exactly why this helps. I'm guessing it's just the S protein is cleaved in S1, S2 by furin. Okay. Well, but they remain connected or associated, right? No? Yes? No? The S protein is cleaved into S1 and S2 by furin. Okay, what's the next picture? Pictures. We love pictures. We're big picture guys here. They're associated, but they're not covalently bonded, allowing for a new function. Structures of the spike protein, its subdomains, and interaction between the RBD and ACE2. Okay. This is the sequence of nucleotides. This 1273, or is this the sequence of amino acids? No, this is the amino acid sequence. That's what this is, right? Okay. And just the sequence for the spike protein. NTD, I don't remember what this is. Probably cringe. Hi, baby. The RBD is your receptor binding domain for whatever all this bullshit is. Loop, this is all cringe. N-terminal domain, no one cares. Okay, A. So this is just the sequence of amino acids. B. 630 loop is disordered. C. Transition to up position is critical for RBD binding to ACE2. 630 loop is ordered and stabilizes the S1, S2. How does it transition from up to 3 down, from 3 down to 1 up? How the fuck does the transition happen? I know the guy mentioned that in the paper as well.